Hello, my name is Jessa Westheimer and I am a Choose Development Fellow. During the summer of 2019, I worked at the Uribe Lab at Rice University on my project entitled Investigating the Role of the Planar Cell Polarity Pathway During Kidney Cystogenesis in Zebrafish. In this video, I will take you through my poster detailing this project. The overall goal of this project is to better understand the planar cell polarity pathway through the study of genes of interest expressed during zebrafish kidney and cilia development. The planar cell polarity pathway is a non-canonical Wnt signaling pathway where core PCP effectors function together to establish polarity in a sheet of cells. One of the genes of interest in this project, prickle1b, expresses the protein prickle, which interacts with transmembrane protein VANGL to mediate PCP signaling as a negative regulator. Previous studies have established inactivation of prickle1 results in kidney cyst formation and cilia with disorganized basal bodies. PCP signaling ensures cells are oriented properly and specific morphogenetic events can take place during development in tissues. One example of this is the patterning of primary cilia. Cilia are hair-like organelles that project from all cell membranes and serve functions in the movement of fluids and signaling. Primary cilia are patterned on cells by the PCP pathway and are regulated by the intraflagellar transport or the IFT pathway. The gene KIF3A, studied in this project, encodes a component of the kinesin motor which shuttles intraflagellar particles up on the cilium to build and maintain it. Our last gene of interest is IFT57, which is a core component of the IFT particle involved with anterograde ciliary transport. Deletions of both of these genes result in severe defects in cilia formation and maintenance, and often are associated with renal cyst formation. Improper ciliogenesis is linked with the formation of cysts on the kidneys and is often a feature of cystic kidney diseases like autosomal dominant polycystic kidney disease, which affects over 12 million people globally. For this study, we use zebrafish embryos as they have been established as a model organism for kidney development. Zebrafish have a pronephros, or basic kidney, which is functionally, structurally, and genetically analogous to the nephron of the human kidney. Additionally, they serve as an excellent model for visualizing cilia in the developing embryo using various assays, including immunostaining. In our first experiment, we tested to see if the genes of interest, prickle1b, IFT57, and KIF3A are expressed at the right time in the right place to influence kidney development. Thus far, we have successfully cloned out prickle1b and have used whole mount in situ hybridization to see that it is expressed in the pronephros at 24 and 48 hours post-fertilization, but expression is lost at 72 hours post-fertilization. In our second experiment, to establish a reliable assay to detect the presence of differentiated cilium, we utilized immunohistochemistry to visualize where alpha-tubulin, a component of cilia, is localized. We were able to visualize strong localization of cilia to the developing olfactory pit in 48 and 72 hours post-fertilization zebrafish embryos using high-resolution confocal microscopy. In Figure 2, we see that alpha-tubulin is specific to the nose region, where we would expect cilia to be expressed. For future study, we will be cloning out KIF3A and IFT57, which are intraflagellar transport genes, and then using expression analyses and whole mount in situ hybridization assays in order to determine if they are also localized to their developing kidney. We will also test if IFT57 and prickle1b genetically function together to coordinate cilium orientation and formation, thereby helping to uncover how potential defects in the IFT and PCP pathways may result in improper ciliogenesis and renal cysts. To test the synergy of these genes, several co-knockdown experiments using CRISPR-Cas9 gene knockout technology will be conducted in different combinations of the three genes to determine if progressive severity of the cystic kidney and disrupted cilia phenotypes occur in the embryo by comparing them to the wild-type data we have presented here. Investigating these genes, which are involved in the planar cell polarity pathway, and the role that this pathway plays in the formation of cilia and the kidney in zebrafish, gives us more context and understanding as to how and why kidney cysts occur, and overall the pathology of cystic kidney diseases like autosomal dominant polycystic kidney disease. Thank you!